I'll just touch briefly upon exercise 2 and exercise 3 here. So what happens if you look at higher diffusion components or eigenvectors using the diffusion map? Do you see anything interesting? No one saw anything interesting? Did anyone try this? So at least when I did it, I didn't really see that much. At least, it's particular for the later time points, it seems very difficult to resolve. So you get a little bit of a split, but not much. And the same thing for exercise 3, that if you use a different subset of genes, it actually doesn't change very much. It seems to be quite robust to what you're using. So we'll move on to the last topic, which is differential expression. So we've been mentioning this several times already, and as you're probably aware of, in bulk RNA-seq, looking for differentially expressed genes is a sort of one of the most common operations that people do to their data sets. So for example, you want to compare a wild type and a mutant, stimulated and unstimulated conditions, and so on and so forth. Turns out that with single-cell RNA-seq, it's actually not that useful or a common scenario to have, where you have sort of two known samples that you want to compare. Typically what happens is that when you get a sample, it is heterogeneous, which means that the first thing that you need to do is to sort of either cluster it or align it according to some pseudotime, and then you end up with either with a continuum or multiple clusters. So in that sense, it typically is not as a sort of interesting operation to carry out as it was in the bulk RNA-seq. And you could even argue that in the first place, that if you have sort of what you believe are sort of known homogeneous groups of cells, there's actually not much point in carrying out single-cell RNA-seq to compare them. You can probably save a bit of money doing bulk RNA-seq instead. But nevertheless, we will take a look at this. And what we're first going to be discussing is synthetic data. So one of the problems, as you probably realized when we're dealing with single-cell data, is that we don't really have the ground truth. And it's very difficult, as we mentioned before, it's very difficult to carry out sort of meaningful validation experiments using orthogonal methods. So one way of dealing with that is, of course, to generate your data synthetically, because then you will know for sure what the ground truth is. The problem is, of course, that you need to make sure that the model that you're using to generate your data is sort of sufficiently realistic and close to what you actually be observing. So there are one application which we won't be covering, but which people have tried, is to sort of generate synthetic reads to evaluate various types of mappers and other types of aspects of computational methods. What we're going to be talking about here is instead to generate essentially synthetic expression matrices. So we have developed some methods for that, which are included in the functions that you're loading. So you can basically run this code here to get those going. So we're going to use something called the Poisson beta model, which is conceptually related to the negative binomial distribution or Poisson gamma model that Davis mentioned yesterday. The idea here is that you're assuming that gene expression is generated by a Poisson distribution where the parameter, the Poisson intensity parameter, is drawn from a beta distribution. And this model actually has quite good biophysical underpinnings because this beta distribution derives from a model where you assume that you have a promoter which switches between an on state and an off state. And so you assume that you have sort of transcription happening at a fixed rate when you're in the on state, and then no transcription when you're in the off state. And because of this random switching, you basically get amplification of the noise or the variability. So this parameter has this distribution has three parameters, which we're going to be calling gamma, alpha, and beta, or K, alpha, and beta. And essentially, you can of course use those parameters to calculate the important properties of this distribution. And in this case, I've listed the mean and the variance of this distribution. So the first thing that you're going to be doing is to explore the different behaviors of this distribution. And to get you started about what's happening, I've shown some examples here, which are the sort of the three types of distribution behaviors that you can observe. So we can have either something which is bimodal, where you have sort of two modes, one of them centered at 0, and the other one sort of further out. It's basically centered at around where the first parameter K is located. And then we have the second type of behavior where it's just a monotonically decreasing. And finally, we have the third type of behavior where we have it sort of just a unimodal distribution. And so your first job is to your first exercise is to try to explore this distribution by modifying these parameters. And as I mentioned, the parameters A and B, the second and third parameter in this model, they are related to the rate at which you're switching between the active and the inactive state of the promoter. So basically, having a high A means that you'll be sort of leaving the you'll be leaving the inactive state at a high rate, and a high B means that you're leaving the active state at a high rate. Whereas the first parameter K corresponds to the rate at which transcripts are produced when you are in the active state. So it basically tells you how fast the promoter is firing once it's on. So you can basically, so you should think of each different parameter combination here as sort of one gene. So and essentially what you're generating here would correspond to a single line or a single row from your expression matrix. Okay, so does that make sense? No questions. So I will let you explore these parameters and get a sense for how this model works. And then before we move on, actually I will move on to the next step here as well. Just to mention that we can also introduce the fourth parameters into this function, which is just simply the number of samples that we're drawing from the distribution. And what you will notice is, of course, that the more samples that you draw, which is, in this case corresponds to the more cells that you're sampling in your experiment, the better or the more accurate your picture of this distribution will become.